Well, the clock on the back wall says it's almost time. The clock on my wrist says we're over time, so I'm going to split the middle and go ahead and start. So good morning. Why don't I start by opening us in prayer, and then we'll get going. Dear Heavenly Father, Lord, thank you that we can be here together in a country and in a city where we are free to come and learn and worship and think and express ourselves. Lord, thank you for One Purpose Weekend and all the wonderful things that we have been able to learn and fellowship over in the last few days. Lord, I pray for this session. God, you are the creator of all things. You are the one who knows all and who can do all. And Lord, our understanding of who you are and what you can do is just scratching the surface, Lord. Lord, I pray today that you can use me to help show some of the details of your truth and your craftsmanship in the way that we are made, in a way that people understand and that can resonate with them. So, Lord, help us think this morning and help us learn and help us appreciate all that you are and all that you've done. We ask this in your name, amen. So for those of you who don't know me, I am Mark Merrick. And it says up here on the screen, I got a whole bunch of letters behind my name. In Philippians 3, Paul explains his resume for people who need the resume explained, where he explains his pedigree as a Jew. And then he goes on in Philippians 3 basically to say that none of that's important. And what was important is that he is a servant of God and that God guides him. So I would really appreciate it if you not worry about all the letters behind my name, unless you need to worry about letters behind my name. And instead, my name is Mark, not Dr. Merrick. I'd be happy to work with you as Mark. So let's kick this off. I have spent pretty much my entire adult life either taking care of patients or treating people how to do so. And I'm an athletic trainer by training, so that's a sports medicine background. But I'm also an academic, and as an academic, we often like to put disclaimer slides up at the beginning of our talks because it keeps us honest and lets you know things you need to know. And in this particular case, professionally and for free speech reasons, disclaimers like this are often really helpful to distinguish when I'm speaking on my own behalf versus when I'm speaking on behalf of somebody else that I represent. Today, I'm speaking on my own behalf. All these ideas are mine and have nothing to do with who I work for or where I work. But here's maybe the better disclaimer. When I started putting this talk together, I did not set out for this to be a talk about creation versus evolution. That was not my intention, really almost didn't cross my mind. What I really wanted to do is show you some of God's handiwork and some of his ingenuity in the amazing and elegant ways that we are designed and built. You are an amazing creation. And we're going to show you just a few examples of that, but there are thousands. But I'll get into a couple. But as I started working my way through this talk, the recurring thing that came up with every example that I'm going to walk you through is, why did that happen? How did that happen? Why are we like that? And as a result, it means that we'll have to talk about the probability that some of these things could have happened randomly. And I think you're going to recognize as we go through that the probability of some of these things happening randomly takes more faith than believe in God. So let's walk our way through a couple of things. I will start with a quote that hangs on my office wall. This is a quote from Francis Bacon, and you may have heard of Francis Bacon. He's been gone a little while. This was originally published in 1625, but he wrote a book called The Essays, and it's a collection of essays. And in one of those essays, called On Studies, he talks about how to study and how to think about things. And this is a quote from that. It's one of my favorites. It says, "Read not to contradict and confute, nor to believe and take for granted, nor to find talk and discourse, but to weigh and consider." Still a few seats up front. There's a few seats on the edges. There's a few here. Looks like we're going to have a full house today. The reason I show you this is I ask you to do what this quote says. I ask you to put aside your preconceived notions, and we all have them. We all have a framework with which we think about the world. And I want you to set that aside, and I want you to simply ask questions of yourself and think, how did this happen? Why did this happen? And enjoy and appreciate some of the elegance that we're going to see. We are naturally inclined to hear information and accept it when it aligns with what we believe, and we're naturally inclined to discard it if it doesn't align with what we already believed. That's one of the frailties of humans. So use this approach as we think about things today. So some origins for this talk. This is a great verse. God said, "Let us make man in our image, after our likeness." So God created man in his own image. In the image of God, he created him, male and female, he created them. I think you are God's greatest creation. And I say that you are God's greatest creation because you are the only creation that he made in his own image. Everything else he made in the image of something else. But you are the only thing he made modeled after himself. And to me, that puts you at the top of the list. I am in awe of the way that he created you, in the way that you work. So I get really geeked out about this stuff, which is probably why I became a professor. But that's okay. I'm going to share a little bit of my geekdom with you today, and you're either going to enjoy it or think, boy, he's really corny. But that's okay. So you are God's greatest creation. Let's start with that. The second thing is this. This is a story from 2 Kings. And in 2 Kings, we have Elisha. Elijah is gone at this point, and Elisha's been given a double helping of Elijah's spirit. And in this particular case, the king of Aram has come up, and he's surrounded a whole bunch of people, including Elisha, and people are very concerned. And they're worried because he's got this big army that's surrounding them, and he's come after Elisha. And Elisha is not concerned at all. And Elisha has a helper named Gehazi. Gehazi is not very bright. He goes on later to actually get leprosy because he's greedy too, but that's a different story. In this particular story, Gehazi's talking to Elisha, and he's really worried about this army, and Elisha's not concerned at all. And Elisha prayed and said, "O Lord, open his eyes that he may see." So the Lord opened the eyes of the young man, and he saw. And behold, the mountain was full of horses and chariots of fire all around Elisha. A lot of times I feel a little bit like Elisha. Not that I've got horses and chariots of fire around me all the time, but because in the way God has made his spirit work in me, I see things that I think other people don't notice. And in this particular case, my hope is that I can turn you into Gehazi for a day, not the leprosy day, but for the day when your eyes were opened and you saw all around you things that are there that you never realized were there. So that's sort of the origin of what I wanted to do with this talk. So I'm going to do this talk in two parts. The first part of this talk is letting you see through my eyes. And I'm going to give you some examples of the way that you are fearfully and wonderfully made. And I thought a lot about how I would approach that because there are so many examples, and we could take a whole semester and do this. That would be a blast. But instead, we have an hour, so I better get moving. So some basic foundations for you. Anatomy and physiology. So by show of hands, how many of you have ever had a human anatomy or human physiology course? All right, a whole bunch of you. I'm sorry. As a guy who's taught anatomy for many, many years, I understand that the way we usually teach it is dry, and it's boring, and it has long lists of stuff you have to remember, like all 26 muscles of the forearm and their innervations and blood supply. Go. Yeah, maybe not. And that's not the way I want you to think about it. Anatomy and physiology is the story of what are our parts and how do they work. And the thing that I want you to come away with is there's a story there, and it's a fascinating, amazing story. So human anatomy is the study, well, any anatomy, is the study of the parts of a structure and the relationship of those parts to each other. And physiology is related. It's the study of the function of that structure. And we can't separate structure from function. Structure and function go together and determine each other. So that's a really key piece of this. So as we look at pieces and their structures, their function is controlled by the way that those structures work. If we break this down a little bit and think about anatomy, we can think about anatomy on what we call a macro level. Or you may have heard the term gross anatomy. Gross anatomy doesn't mean that it's gross to look at. It just means that it's big. And gross anatomy is the anatomy that we can see with our naked eyes. So it's large structures visible to the eye. Now sometimes we have to remove skin and muscle and fat and other things to see them, but you could see them with your eye if we got deep enough. We also have microanatomy, which are things you can't see with your eye. They're too small, doesn't mean they're not there, but they are too small. In many cases, that microanatomy is more fascinating than the macroanatomy. We love to talk about examples of macroanatomy because everybody can identify with them. But the microanatomy tells an even richer story. And microanatomy usually is the study of things like cells, which we call cytology, or tissues, which we call histology. And tissues are just collections of cells that have similar functions. Another layer deeper is biochemistry. How many of you have ever had a biochemistry class? A couple of you. Very good. So biochemistry is the chemistry of life. Life happens at the subcellular level. Life is tinier than tiny. And it happens because you have substances that do things, and you have subcellular machines, molecules, that work on those substances to make them do those things. So you have tiny little microscopic machines inside of your cells. And that's really what biochemistry is. I once had a professor who explained to me that life is biology. Biology is really chemistry. And if we wanted to go a layer deeper than that, chemistry is actually physics. But we're not going to do that piece because there's places I just won't go with you this morning. So those are some foundations. So with that as a framework, let's talk about a few examples. The first example I want to talk about today is the macroanatomy of redundant blood supply, which sounds like a really boring topic until you figure out what it is and what it will do for you. So most of you, I think probably all of you this morning, have an upper extremity. Has your hand ever gone to sleep? Of course it has. Usually when something goes to sleep, it goes to sleep because we have pinched off the blood supply for a little bit too long, and we've run out of having enough oxygen in the area, and we haven't cleaned out enough waste. Maybe the starting point for this is everything in your body runs on a battery, and that battery is in a phosphate bond. That phosphate bond simply means I stick a phosphate on something and there's energy stored up in that bond, and later on I'll snip that bond and release the energy that I can use it. And that usually happens in a molecule called adenosine triphosphate, and I'm not going to go through ATP with you today. But what you need to know is that for me to make those phosphate bonds, for me to have energy that I can store in a form my body can use, I pretty much need oxygen to do that. And when I run out of oxygen, my ability to make that kind of runs out, and as a result, my cellular functions cease. If I go without oxygen as an organism too long, I don't survive. You may have even heard of something called the rule of fours before. You can live about 4 minutes without oxygen, about 4 days without water, and about 4 weeks without food. Or for some of us, we're going to make it a little more than 4 because we've been storing up. Oxygen's important. And the way that you get oxygen anywhere is you pump it around through tubes in your body called arteries. We have a blood supply. That's not a new idea for you. What you've probably never looked at before is how does your blood supply work? So there's a recurring pattern in your extremities, and I'll walk you through that. So this is a map of the blood supply of the upper extremity. And I know it's a little bit small. Wish we had giant screens, but this is as close as we're going to get. So here's what you really need to know. In the upper segment, the upper section of a limb, you have something called a conduit artery. A conduit artery is simply a tube that carries stuff from one place to another. It's a hose. Conduit arteries' jobs is not to feed anything directly. They're the big supply pipe. And I have little pipes that come off of that supply pipe and then feed other structures as we need them. So in any extremity, whether this is your upper or lower extremity, in the proximal limb, the proximal segment, the part closest to your body, one conduit artery. Then in the lower segment, below the big joint in the middle of the limb, you have two conduit arteries. In the arm, the conduit artery is called the brachial artery in the upper segment. It actually has multiple names. Brachial artery in the upper arm, axillary artery in your armpit, subclavian artery in your upper chest. It's all the same artery. It's like a street that changes name every blocks. Still the same pipe. It comes down to your elbow and it divides. And when it divides into two conduit arteries, your radial and ulnar artery. And the radial and ulnar artery, there's one coming down each side of your arm. And if you've ever taken your pulse at your wrist, that's your radial pulse. You're feeling the blood pumping through that radial artery. Now, at the end of those two conduit arteries, you have two arterial arches. And an arterial arch is where those arteries join together. And when we join two tubes together so things can flow between them, there's a word we use for that in anatomy called anastomosis. So there's a big word for you today. It means I connected two hoses. Simple idea, big words. Welcome to academia. So I have two arterial arches in the hand. So let's take a look at those. This is the blood supply of the hand. And the things you'll notice in this blood supply of the hand is first that there's those two conduit arteries, the radial artery and the ulnar artery. And as they come into your hand, they connect to each other. They make that anastomosis. In this case, it's one artery connecting to another, so we call it an arterio-arterial anastomosis. Another big concept, simple idea. We also sometimes have them where an artery connects to a vein, like through a capillary, and we call that an arteriovenous anastomosis. In this case, you have not one, but actually two. You have a deep one and a superficial one. So the question is why? Why would you have this? Why would you not simply have branches that come off of each of those arteries to feed individual structures? Why do they connect to each other? And the answer to that is called redundant blood supply. And the idea of redundant blood supply means that if I lean on one of them and I pinch it off for a period of time, I still have blood supply coming through the other one that can feed everything that needs fed. So we're going to do a little laboratory experiment here this morning, and I'm going to show you how this works. It's a really easy thing for you to do. So what happens when you block the conduit arteries? So to do this, if you hold up your arm, you have an artery on each side. And I'm going to have my wife come up here and hold this mic because I have only two hands and not three. So if you take your hand, you reach up from the bottom and wrap your thumb and like your middle finger around and pinch these two sides of your wrist. So just like this. And you squeeze your hand tight. When you do that, and you open your hand will get really pale because you've just cut off all the blood supply to your hand. Now, if you lift your thumb or your middle finger, watch what happens to your hand. Maybe you can see this in mine. Did you see it pink up? It pinked up because we restored the blood supply. Now you can do that again. Pinch it off again. This time open the other side and see what you see. Thank you, dear. So what happens? No matter which side I open, my hand pinks back up, right? That's because I have a redundant blood supply. Is that a question in the back there? Okay, no worries. If you have a question, just ask. We're all among friends here. That redundant blood supply is handy because it turns out we use our hands for everything. I am twisting, I'm turning, I'm gripping, I'm doing a lot of different things, and I get in positions where I pinch off one of those arteries all the time, and my hand would very quickly fail if I didn't have that redundant blood supply. That is an amazingly cool design feature of the way that you are built. And guess what? It's not just in your hand. It's in every joint in your body. It's in multiple of your organs. And you have a really elaborate one in the middle of your brain called the circle of Willis, where you have multiple arteries that come together and feed this ring that then feeds the brain. So if I pinch off either side, I still have blood supply, which is really helpful because my brain needs all the blood it can get. So here's another question for you. Did you notice in this diagram where the arteries to the fingers are? Where are the arteries on your fingers? Are they on the palm of your fingers or are they on the sides? They're on the sides. Why? What would happen if the blood supply to my fingers came up the palmar side? What would happen if I did this? I would trip. If I carry my briefcase, guess what happens to my blood supply if it's in the palm of my fingers? I lose blood supply, don't I? So how cool is it that your blood supply to your fingers is on the sides where they don't get compressed? This is the stuff that geeks me out, because my question is, why is that there? We can see why it's there. How did it happen? In a world where we believe things like random variation or random mutation and natural selection would work, it's hard to explain this. Because I'm not sure you would die and wouldn't survive to reproduce simply because you had an artery in the palm of your fingers. That's okay, simple example. Let's move on to another one. The macroanatomy of your grip. I'm very hand-focused today. I didn't realize that until I put these slides together. I probably should have looked at another area, should have done the last one in your foot, but that's okay. So if we look at your hand might be one of the most elegant inventions in all of creation. That's a lot to say, so let's unpack that. So when we think about your hand is different than the hand of any other creature that God made. There are similarities to some of his other creatures. Many creatures have 5 digits on the end of their appendage. Many of them have multiple joints so we can move them, but your thumb makes you unique. You have a thumb that is positioned in a way that lets you do things nobody else can. You also have the dexterity in your hand and the strength in your hand to do everything from crushing a can to picking up a fragile little piece of something without breaking it. You can pick up an eggshell and not break it with the same hand that you can give somebody a bone-crushing handshake with that they'll never forget. So your hand can do a lot of things. Now, when we think about how a hand moves, to be able to do all the things that you do with your hand, it has a set of different movements that can happen at each joint. So we have things like flexion, which is where I flex or bend my fingers. We have things like extension, where I can straighten them back out again. We have things where I can spread my fingers. We call that abduction, and we can bring them back together. We call that adduction. That's great. Fingers can do all those things, and they can also spin around in circles, which is kind of cool. Now, I get geeked out when I think that's cool. Look at your thumb. So look at your hand, put your thumb up. Think about how your thumb moves. Your thumb can flex, that is, it goes up and down. If you put your fingernail side up, it's the easiest way to orient yourself and see these. So thumb flexion, up is down, extension is up. I can abduct my thumb, and I can bring it out this way, like I'm grabbing a can. I can adduct my thumb and bring it in this way, like I'm pinching something between my thumb and first finger. I can also reach my thumb way across over here and touch the other side of my hand, and we call that opposition. The ability to do that means you can do a lot of things with your hand. As you look at those hands, we've got bones through the palm of your hand, and those bones in the palm of your hand, the 2 on your little finger side, move. The other 2 don't. So when I do this and curl my hand like I'm going to carry something, then I rotate a couple of bones in my hand. Very elegant, the kind of small movements that you put together to make your hand move. So the question for you is, how does that work? Well, I'm only going to go through a piece of that because we could spend 2 weeks on the hand, and I'd probably enjoy it a lot more than you. So when we look at the hand, I want to look at how your fingers flex, because it's really, really unusual and kind of fun. So there are 3 muscles that cause your fingers to flex. And they have big fancy anatomy names like flexor digitorum superficialis. What do you think a flexor digitorum would do? Flex the digits, right? There you go. And superficialis probably means it's superficial, close to the surface. There's another word in anatomy that's the opposite of superficial, and that's profound or profunda. So we also have a flexor digitorum profundus. Flexes the digits, it's deeper. So the names tell you where they are and what they do. That's a cool thing about anatomy. It's got like secret code. The last one, I can't help you with the name a whole lot. It's called the lumbrical. And the lumbricals are in your hand. So the way that this works, if I forward this, so I have highlighted these in color here for you. So we'll start with the flexor digitorum superficialis. This is a muscle in your forearm that causes movement in your fingers. So it has a long tendon, crosses your wrist, crosses through your hand, and goes all the way down in your finger. And as you look at that all the way down in your finger, the superficialis is the green one here. I would think the superficial one would be the one that goes all the way to the end. And I'd be wrong. The superficialis stops. If you look at your finger, you've got 3 bones in your finger. The superficialis stops on the middle bone. I also have the profundus, also big muscle in your forearm, long tendon, goes across your wrist, down into your hand, all the way to your finger to make your finger flex. The profundus goes all the way to the tip. For that to happen, those 2 tendons have to cross in your finger. Now there's a third one in here. I mentioned the lumbrical. The lumbrical is actually in the palm of your hand, and it's between the bones of your hand. If you've ever worked with somebody who has like that bone-crushing handshake, who works with their hands, think of like a bricklayer, they got big, meaty mitts. They got really well developed here because they're going to do a lot of grip work. The lumbrical, the one that's closest, only goes to the very first bone, the bone furthest from the end. So all 3 of these tendons come in and they cross in your finger, and they let you do something remarkable. If all I had was lumbricals, I could do this. They only flex the joint where your finger comes to your hand. Now, if I look next at the superficialis adds to that movement because it crosses that joint, but it also flexes at my middle finger, or at the joint in the middle of my finger. And the profundus goes all the way to the tip. So it helps here, and it helps here, and it lets me curl my fingertips. Your ability to curl your fingertips is a spectacular thing, a spectacular function of your hand that happens because of the way all of this is built. So the question that I would ask, by the way, these are all strapped down with other straps that anchor them in place, and they allow you to do very delicate motions of the hand. Why and how would that happen? Well, the why is pretty important. You ever broken a finger or been in a splint and try to use that hand? Good luck. You're like flapping around, you can't grip things, you can't hold a pencil, it's tough to feed yourself. This lets you do all those things. The why is there, but how would you develop that? How would you randomly get these 2 tendons to cross and at the same time have all the anchors that come around them that allow you to move discreetly, one little segment by one little segment? Pretty hard to explain how I would come up with that. So again, really cool example of how the body works. Now, that's enough on the macro examples, because macro examples are big and they're easy to see. The good stuff is underneath. So, example number 3, microanatomy of the skin epithelium. Another big word, like a lot of big anatomy words today. So, your whole body, all of the parts of you, are made out of just 4 kinds of tissue. Excuse me. So everything in your body is either nervous tissue, which you can pretty much figure out what nervous tissue does. Conducts signals, nerves, you get the idea. Muscle tissue contracts and moves. We have like skeletal muscle, we also have cardiac muscle, your heart's made of muscle, and we also have smooth muscle in places like your blood vessels that make them dilate or constrict. So muscle makes things move. We have connective tissue there in the bottom right, and connective tissue is structure and reinforcement. So, lots of things there. They support, protect, transport, storage. So you get things like fat, but you get a lot of things like bone and tendon and ligaments and cartilage are all in the connective tissue. And then we have the coolest tissue of all. Well, maybe. People can argue on this. Epithelium. I am fascinated by epithelial tissue. Epithelium, it says here, is protection, absorption, secretion, filtration, sensation. Sensation's probably a little more nervous, but we could debate that. So epithelial tissue is interesting because epithelium serves as a covering or a lining everywhere in your body. So as we look at your body, there's different kinds of epithelial tissue. We have epithelial tissue. I've got tiny little things on here, so I'll try to read them. So we'll start here with A, and we have simple squamous epithelium in the air sacs of your lungs. We sometimes use words to describe shapes. Squamous means shaped like a pancake. So we have simple pancake epithelial cells, and this lines the air sacs of your lung. They're a single layer thick, which allows oxygen from the air to diffuse through them and get to your blood supply. If they were too thick, it wouldn't work. But there's a reason that it's epithelium and not something else, and I'll circle back to that. We have epithelium that lines places like the tubes of your kidneys, or that lines every blood vessel that you have. Every blood vessel that you have, the inner lining is epithelial tissue. We have we have simple columnar. Columnar means it's shaped like a column. They're tall and skinny. So it's like a stack of cans next to each other. Columnar epithelium is in the intestine. And we absorb things through that you've eaten. We also have, as we work our way around here, some pseudostratified ciliated columnar epithelium. That's a mouthful. And it says respiratory tract. The epithelium in your respiratory tract is very cool. Ciliated means it has little hairs on it that act like filters or combs. And in your respiratory tract, as you breathe in, you are breathing in all the junk in the air around you. You ever stood in the sunbeam and seen all the little particles of dust illuminated in the air? Guess what happens to those when you breathe? In they go. They don't come back out. They get trapped in the mucus inside your respiratory tract, and those cilia move that mucus along up your throat, and then we clear our throat. And then clear our throat, and we swallow it down to the acid bath in our stomach to kill it all. So really handy. Guess what smoking does to these? Smoking destroys them. And as a result, I don't have those ciliated cells anymore, I don't clear stuff out as much, and smokers have a much higher frequency of things like respiratory infections for that reason. This is one of the bad things about smoking. We're not here to talk about smoking today, but that's what it does. And the last one that's on here is stratified squamous epithelium. Stratified means it's in layers. And squamous, we said, means shaped like a pancake. So I have pancake-shaped stuff stacked up on top of each other. This one, it mentions esophagus, but then not only is that in the esophagus, that's also your skin. Why is epithelium in all of these different places? Well, the answer is it grows back really, really well. Unlike nervous tissue, when you damage it, it doesn't really come back. If you've probably seen if somebody's had a nerve injury or if somebody has a stroke, there are things that never come back. Nervous tissue is very difficult to replace. Muscle tissue, not as hard as nervous tissue, but still very difficult to replace. So when you've had a muscle tear and it heals up, it didn't heal up by growing new muscle. We glued it back together with scar tissue, and it's never quite as strong as it used to be. Connective tissue heals better still than muscle. Bones will heal up. But when you break your bone when you're really young, it heals fast. Toddlers bounce. Their bones are mostly kind of green and cartilaginous, and if they do get a fracture, they're healed up in 2 weeks. If you're in your 70s or 80s and you get a fracture, it's not 2 weeks, is it? It's 8 weeks or 12 weeks or 16 weeks. And part of that is the cells that cause you to heal. You have fewer and fewer of them as you age. And then there's epithelium. Epithelium heals fast. It is designed in such a way that it is constantly reproducing itself. And because it's constantly reproducing itself, it heals really well. So guess where it's located? All the places that have exposure to the outside world are covered in epithelium because it heals so well. The outside world damages my tissues all the time. You are shedding cells on the inside of your clothes. You're leaving cells on your chairs. This sounds gross. This is where the dust came from that we already breathed in. More gross. But you're replacing that all the time. So stratified squamous epithelium I mentioned is in the skin. So let's take a look at that. Oh, fun fact for you. All the parts of your body that you can normally see are dead. You can't see any of your living cells. Everything that you see, your skin, your hair, your nails, the lining of your GI and respiratory tract, the surface of your eye, are all covered with epithelium that's dead. So here's how that works. Here we go, histology slides. Never thought you'd have histology on a Sunday morning. So if you think about that statement for a minute, that's a great thing to build into our system, that the outer surface is already dead, and we're already in the process of shedding it and replacing it from underneath before it ever got to that outer surface. That's a really great design feature. It gives us the ability to heal. It gives us the ability to survive. And when we look at the skin, the outer layer of the skin is called the epidermis. And that epidermis has multiple layers. It has 5 layers. Sometimes it has a 6th layer in the palms of your hands or the soles of your feet where the skin is really thick. But if we start at the bottom, you'll see dermis. Dermis is where your blood supply is, a bunch of living cells in your dermis. Above the dermis, we have something called the stratum basale. The stratum basale is the first layer of the epithelial part of your skin, the most outer part. It's alive, and it's dividing constantly. So those cells divide to make new cells. And when it divides, the old cell gets pushed one row further away from the dermis. I grow the new cell underneath it. And then as it divides again, that other cell pushed another row further away from the dermis. So as these cells keep dividing, I keep pushing the older cells further and further away from the dermis. The dermis is where the blood supply is. So what happens to my cells when I get them too far away from the blood supply and too far away from the oxygen? They die. And this is by design. This is by design. So as I work my way up, we have that stratum basale where they're dividing. I have a layer above it called the stratum spinosum. And the stratum spinosum, they start to dehydrate a little bit, and you start growing a whole bunch of protein in them called keratin. Keratin's what makes your skin waterproof. It's also in a lot of cosmetic products. You've probably heard the word before. As it pushes further away, we get to a layer called the granulosum. The granulosum layer is full of granules. That's what the name means. If I look at them under a microscope and stain them, there's a bunch of little spots on them. And that's as those cells dehydrate and lose their fluid, all that protein in the keratin that makes them waterproof collects stain, and that's why you see the colors. So we have the granulosum layer. And then above the granulosum layer, sometimes you have a stratum lucidum. That's a clear layer in the palm of your hands or soles of your feet where the skin's thick. It's not in other parts of your skin. And then I get to the very outer layer, the stratum corneum. As you look at the cells there in the diagram on the right, at the basale layer, the cells are kind of like little cubes. And every layer that they get further away as they dehydrate, they get flatter and flatter and flatter, till eventually, as you get up there and you get up to the corneum, well, you see even in the granulosum, they now look like bricks because they dehydrated out and gotten smaller and thinner. And then eventually in the stratum corneum, no more oxygen, they've lost all their fluid, or most of their fluid. They're dry, and they're little flakes, little pancake-shaped flakes. And they're all stuck together. And if we were to, you can even do this, take a fingernail, scratch it along the back of your hand, and you'll see a little bit of white residue there. You just scraped up a bunch of your little pancake cells off of the skin. The advantage of doing this is I'm always shedding those. So when I cut myself, most of the time the cuts are in the epidermis. They may get a little bit deeper, but they seal up pretty quick. But I've already grown new skin underneath it that's on the way, which lets me heal. So that's great in the skin. Would that be handy in the lining of my lungs? You ever breathe something really hot? You can damage your lung tissue. Ever get an infection? We shed some of that stuff off, we cough it up. Would that be handy in the lining of my GI tract, where I'm pushing foodstuffs through? Sometimes we eat crunchy things. And crunchy things scrape along, and I need to grow new cells underneath. So the whole idea of epithelium is I grow new stuff. And by growing new stuff, I repair the damage. So these are designed in a way that all the places that come in contact with the outside world are designed to shed the damage they get from that outside world and replace themselves quickly. That is the beauty of epithelial tissue. Amazing function. So how would that happen randomly? For that to happen randomly and for me to have epithelium in so many different places, that change would have had to have happened really, really early in the process of development. And then it gets distributed to multiple places. But yet, in each of those different places, it takes a different form. Same concept, but different form. This is getting really complex to make this thing happen randomly. Recurring theme here. All right, buckle up, here we go. Biochemistry of proteins. You're now on upper level understanding of this stuff. And again, every layer we get a little deeper, the more fascinating this becomes, if you're a geek like me. But this is enzymes and protein biochemistry for non-scientists. So you do not have to have a science background to be able to understand. We're going to just walk you through how this works. So the first thing you need to understand is that enzymes are really, really important. And you've probably heard of enzymes. You hear about, there are enzymes in different foods. You don't want to kill, if you overcook things, you kill the enzymes. What is an enzyme? Well, an enzyme is a specialized protein, and I'll talk about what a protein is in a minute. In some cases it's RNA, but usually it's a protein, and it acts as a catalyst in a living organism. So, catalyst means it helps a chemical reaction happen. So it catalyzes that reaction. In this case, the way that an enzyme works is the things that are going through a chemical reaction we call substrates. And in chemical reactions, we do basically two things with substrates. We either take two or more things and stick them together, or we take one thing and split it apart. Enzymes catalyze that reaction so that it happens. And that's really, really important. And they do that by holding the substrates in exactly the right position. So the right parts of them are near touching each other and can react with each other. And that's important because these substrates are sometimes big and they sometimes have complex shapes. So imagine that I have two Legos. Those two Legos are substrates. Imagine I take those two Legos and I drop them in a bag and I shake it up. What is the likelihood that those two Legos just linked to each other? Pretty small, right? Are they designed to link to each other? Absolutely. If I hold them in the right place and push, will they link to each other? Absolutely. But the probability of it happening by itself, really, really small. Now, an enzyme is like your hand and the Legos. It holds those Legos, holds the substrates in the right position so the right parts are touching, and typically it uses energy in some format, usually that phosphate that I talked about before from the phosphate bond, and it either pushes them together or pulls them apart. Pretty simple idea. That's what an enzyme does. It reduces the activation energy needed to make the reaction happen. The warmer things are, the faster reactions happen. But in the real world, when I put my two Legos in the bag, there's not enough energy and alignment to make it work. An enzyme makes it work with much less energy and at a much faster rate. It speeds up the reaction. Usually, it speeds it up by a factor of greater than 1,000,000 times. 1,000,000 times faster if I have an enzyme versus if I don't have an enzyme for that reaction. Here's where that's important. None of you are here today without enzymes, because those reactions do not happen fast enough in the wild without an enzyme to be able to sustain life. You have to produce energy and produce a bunch of other things and break other things down and do a lot of it really fast in order to stay alive. Without enzymes, there is no life, period. Life cannot happen without enzymes. The math won't work. The math won't math. How's that? So that's what enzymes do. Without enzymes, reactions are too slow for life to happen. All right, deep breath. How does this happen? So it happens because enzymes are proteins. And I'm going to start here. This is an amino acid. You've probably heard the word amino acid. There are 20 of them. And the amino acids are different building blocks. Think of them as things like Legos. And there are 20 different shaped Legos. As I look at the structure of an amino acid, on one end of it, you see that it's got a nitrogen and 2 hydrogens. We call that the amino end. That's where the name amino acid comes from. The other end has a carbon, 2 oxygens, and a hydrogen. In organic chemistry, we call that a carboxyl group. And a carboxyl is acidic. That's where we get the name acid. So an amino acid has an amino end, it has an acid end, and in the middle, you see an R. That R is stick a Lego here. And there are 20 different R's that I can attach to this structure, and every one of them is a different amino acid. So I make an amino acid by having this structure with one of my 20 R's on it. A single amino acid doesn't do a hell of a lot, but I can link them together. And if I link 2 amino acids together, which is what you get in this diagram, you see in the upper part of it that the hydrogens and the oxygens are kind of having this orange cloud around them. That cloud is an affinity for bonding together. And if I have an enzyme that holds them in the right place and gives them a little nudge, that will come together and it will form what you see in that bottom thing in green called a peptide bond. A peptide bond means that I've hooked 2 amino acids together. And if as we look at this, we would call that molecule a dipeptide, meaning it has 2 pieces hooked together by a peptide bond. What happens if I add another one? Well, at the far end of that second amino acid, I can do it again. And I can hook another amino acid on. And now I have a tripeptide. And I can do it again, and again, and again, and again. And when I get these big numbers of them hooked together in chains, we call them polypeptides. Polypeptides are proteins. So all a protein is a bunch of amino acids all hooked together. But here's the really interesting thing. The sequence of the amino acids makes a difference. Those R groups all have different shapes, and they have different molecules or different atoms that are on them. And depending on which one I put in which place, they have a desire to bond with and link up with other pieces. And that causes this thing to fold. So instead of one long chain, I get what looks like a wadded up ball of string. And the shape is everything. So protein structure. If that upper list we sometimes call primary structure, that's what is the order of the amino acids in my protein. And we can have a code and we can write it out and say that, I've got an alanine here and I have a cytosine here, and it's a list of what order they're in. It doesn't tell you what shape it will take. The next piece is that those things will have an affinity for each other. For example, I have an affinity for my beautiful wife here in the front row. And given any other opportunity, instead of standing up here, I would be sitting right there. And if I'm sitting right there, I'm probably holding her hand, or she's probably leaning against me. Some of you are doing that right now. These molecules want to do that. And when they do, it causes them to twist or to fold or to bend, and it gives them shapes. And as those things shape together, we get these really complex 3-dimensional shapes that are for a protein. So I'm going to go one step further than just showing you that and show it to you what it really looks like in real space. So on the left is a map of how a protein might fold. Now that particular protein We talk about how many residues are in a protein. That's how many amino acids. And they're usually somewhere between 62 and 2,500. That one's probably, about 1,000 residues or so, and it's got different pieces and it's complex. The middle diagram is what it really looks like in 3 dimensions because, there's little atoms on here, and it takes a shape and has a space. And you notice that it says active site. The folding of that protein gives it a very specific shape, and guess what happens in that active site? Well, look at the far diagram. Those substrates, those Legos that we want to stick together, are exactly the shape of those active sites. Exactly the shape. We call it a lock and key fit. So I will have a protein, which is a giant molecule, by the way, big giant molecule, holds little tiny substrates with a lock and key fit, holds them in just the right place and gives them the push to stick my Legos together. This is how a protein works. This is how an enzyme works. So as we think about that, every biologic process in your body uses these enzymes to make things happen. And here's where it gets fun. They have to happen in specific orders. So the process that I have here, and no, there won't be a test on this later, this is energy. This is taking a glucose molecule. You've all heard of blood glucose. Glucose is a 6-carbon sugar molecule. It's the basic building block of energy. This is how I take that and turn it into those phosphate bonds I talked about earlier, which is the form of energy in a battery that your body can use. So on this diagram, every single straight arrow represents a different enzyme. So you see I've got a glucose, and then I have a glucose 6-phosphate up there in the top left. There's an enzyme called glucose 6-phosphatase. Wow, cool name. It sticks a phosphate on it, keeps the thing from escaping, then we break it into 2 3-carbon sugars, and then we break each of those down into bigger pieces, collect all the parts, put them together, and build a battery. That's energy metabolism. The problem is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, maybe 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22 steps in doing this. Each of those steps has its own enzyme. Each of those enzymes is probably hundreds to thousands of amino acids long. And if I get one amino acid out of sequence, the shape doesn't work and it won't work. So I have to have a system where I can make these 21 enzymes. I can make them reproducibly, accurately, over and over again. I have to get the code right, and the code is like a book. So if I have a typo in my book, my amino acid doesn't work and my enzyme doesn't work. And if one enzyme doesn't work, guess what happens to the whole rest of this process? Failed. Won't work. All the pieces have to work for me to get energy at the end of this to make my cells work. Welcome to God's fingerprints in your body. So as we think about that, how did this stuff happen? I don't know. I don't know. And that's part of how I know that God's here. Because we're trying hard and we can't figure these out. But let me walk you through a couple of things. So earlier I gave you that Francis Bacon quote. Read not to contradict or confute, you get the idea. Be open-minded and look at what's actually in front of you and ask questions. How did this happen and why did it happen? So, here's your answer. The Lord God formed the man of dust from the ground and breathed breath into his nostrils, the breath of life, and the man became a living creature. So what is the dust of the ground? It's all the elements on your periodic table. That's the dust of the ground. It's actually not the dust of the ground, it's the dust of the universe. It is God's building blocks. It's God's Legos. And they assemble them in lots of different ways. But you know what? I can assemble those Legos. I can be a synthetic biochemist, and I can make some really cool molecules. And I can't make life happen. The breath of life was more than just having the dust of the ground. You had to have dust of the ground, but that breath of life has to have two things. It has to have the extensive list of all the biological processes and the machinery you need to run each of them. By the way, the number of biological processes in your bodies is numbers in the tens of thousands, perhaps the hundreds of thousands. Each of those processes, multiple enzymatic steps. Each of those with complex proteins that have to be formed in exactly the right way to make them work. You're getting the magnitude of this? Wow. So you have to have all those things, and you still have to have the magic ingredient that we do not understand, and that is the spark of life that turns them on. We can make them, we can't make them work, and that is amazing. So, Bob Ross, pastor referred to Bob Ross. Bob Ross often talked about happy little accidents. So, evolution as a theory. Is evolution through random mutation and natural selection true? That's not a good question. Just not a good question. You have to say true for what? And in this case, is it true as a mechanism for variation? And I think the answer to that is absolutely true. Evolution changes and allows us to, it's a mechanism by which we vary and adapt in some places. And we've seen that in some places, like the shape of the beaks on the finches on the Galapagos with Darwin. We've seen that with the moth who changes colors. It explains things on a particular level, but it is not adequate as an explanation for the origin of life. It is a mechanism by which existing life has small change, not how did we get the life in the first place. And that's where it falls apart. So let's walk through this. Evolution, as an explanation for the origin of life, has several major assumptions. The first thing is that spark of life, that life comes from non-life. We are trying really hard, and we are failing miserably because we can't make this work. We have no examples documenting that life has ever arisen from non-life. None. None historically, none in the laboratory, none in places where we put together ideal conditions, put together all the pre-built machinery of life. Try to zap it with some electricity, it doesn't come alive. There's no Dr. Frankenstein here. Not happening. We have not succeeded in making it happen in controlled, ideal conditions, let alone having it happen in the uncontrolled conditions of the real world. I can control things in the laboratory and get things to happen that don't happen in the real world because I control the environment. In the real world, it's even less controlled. And even in our controlled environment, it's not working. In fact, as we do this, we are struggling to get basic building blocks to spontaneously happen. So I mentioned that we have those amino acids. Well, to build the amino acids and assemble them in a protein, we use DNA as your storage area for your blueprint. We make a copy of that DNA called RNA, and we use that RNA copy to then assemble the amino acids in the right order. Well, those storage of blueprints use things called nucleotides. You've probably heard of words like adenosine. You've probably seen a bunch of these if you've ever studied DNA. We are trying to make nucleotides in the laboratory without really sophisticated synthetic chemistry. And we can't get them to work. We can't get them to assemble. Now, the nucleotide is one piece of the blueprint that I then use to assemble the amino acids already built together in a specific sequence to make an enzyme, to put together in a specific sequence to make a biologic process that allows you to live. So we're 5 or 6 steps removed on the more basic level, and we can't even get that to work, let alone the next levels of assembling them into what they become. Excuse me. The next one, we're really short on time because I'm a professor and I talk too much. That all forms of life today that we have today derive from single-celled life forms. That's an assumption that you would have to make. Here's the problem. We have single-celled life. We have amoebas, we have bacteria, we have a bunch of single-celled life forms. You know what we don't have? 2-celled life forms, or 3, or 4, or 5. We have single or millions. We have nothing in the middle. That's a bridge that's really hard to cross. Nothing in between. That time and chance drives evolution forward. That we have enough of those happy little accidents along the way that they can aggregate and turn into what we need them to turn into. But that requires that small random changes aggregate together in specific ways that make complex things that do very complex interactions further on. The likelihood of that happening is something like, you've all seen alphabet blocks for your kids or your grandkids. What if I happen to throw a few thousand blocks into a bag, shake them up, dump them out on the floor? What's the likelihood that I get a full sentence? That's what we're talking about here. Aggregating little random pieces into complex systems that then work. Really hard to talk me into the fact that that would happen. And the last one on here, that minor genetic variation in a species that we observe is observable evidence of macroevolution. So the finch's beak sizes change on which side of the island you're on is very different than an amoeba eventually became an elephant. Variations within an existing kind of an organism are small changes, and I absolutely believe that evolution does that. It's a mechanism by which those small changes happen. It doesn't come close to explaining how I get the big stuff to happen, and there's not enough time in the world, even with the old Earth models, to get any of this stuff to happen. So there's a book outside that if you're geeked out by this stuff like me, that you might enjoy reading. It's called Darwin's Black Book. It is written by a biochemistry professor, and it's not written for professors. It's written for everyday people. And in that book, he makes a few arguments. The first argument that he makes is something called irreducible complexity. That's what we're seeing and what we've been talking about here today. The idea that systems with well-matched, interacting, interdependent parts that create all of our basic functions happened is astronomically small that could happen. You know, that's not like throwing blocks in and getting a sentence. That's like throwing a whole bunch of blocks in and getting War and Peace with the punctuation right. Just not realistically going to happen. And here's the other really cool thing. These systems are so complex that the whole thing had to happen at once. It's not stepwise because each step along the way isn't functional by itself. They're reliant on other processes at the same time. So any one of those enzymes in that 21 enzyme piece, maybe it evolves. But without the other 20 enzymes, it goes nowhere, and it gives you no advantage that would cause it to be preserved through natural selection. So too complex. The second thing is Darwin's mechanisms are limited. Random mutation and natural selection, as I've already talked about with the finches, can explain some macro level changes within already existing organisms, but it's not enough to get you to the origins. The cell is a black box. That's where the title of this particular book comes from. Darwin, in his time, thought cells were undifferentiated globs of stuff. Had no idea that there was a whole subcellular machinery within them. Could not conceive of that, and it kind of blows his theory apart. It's more complex than by orders of magnitude than anything he ever imagined. Intelligent design is the inference that this book draws. This is one of the first books to talk about intelligent design. It's sort of the origin of some of those conversations. And it concludes that the only way we get to systems this complex is they don't happen spontaneously. They had to be designed from somewhere. Now, this author doesn't say it's God. He just said it had to come from somewhere. Draw your own inference. And the last thing is scientific orthodoxy. The scientific community is reluctant to consider other explanations. See previous Francis Bacon quote. We read to refute rather than reading or listening to understand. So with that said, our happy little accidents, they're not enough. They just are not mathematically possible to be enough. Instead of happy little accidents, you're looking at the fingerprints of God at the subcellular level. So with that said, here's where our title came from. Going full circle, this is in Psalm 139. I praise you, for I am fearfully and wonderfully made. Wonderful are your works. Amen. My soul knows it very well. And I would say so does the rest of your insides, even if your conscious isn't aware of it. So thank you very much. Hope you found this interesting.